Hello students. Before starting the discussion on the disorders of the immune system, let's have an overview of the basic mechanism by which the immune system governs. So the type of immunity. So we have basically two type of immunity, innate immunity and acquired immunity. Innate immunity is something which is present since birth, and its spectrum is broad since it is non-antigenic. It is not stimulated by any antigen, but it is present since birth. So what all cells comes under this immune backup? Neutrophils, macrophages, natural killer cells. And skin is the largest organ which acts as a shield against all type of agents, physical agents, microbial agents. Then comes on to the acquired immunity. Acquired immunity is something, as the name suggests, it is acquired on stimulation by microbe or microbial antigens. And what all cells take part in the acquired immunity are the lymphocytes. So the lymphocyte can be T lymphocyte or the B lymphocyte. So what are T lymphocytes? T lymphocytes, they constitute about 60 to 70% of the circulating lymphocytes, and they are predominantly found in the paracortex of the lymph node. So with that, I'll give an overview of the structure of a lymph node. A lymph node is composed of a cortical area, the cortical area, which is composed of these primary follicles and secondary follicles, which have predominantly B cells. So the primary follicles are those which don't have any germinal center, and secondary follicle are the one which have a germinal center in them. So this is a cortical part, and there is a paracortical part, this area, and this area is rich in T cells, and then there is this medullary part which is rich in plasma cells. So let's get back to where we were. So the most common sites for T lymphocytes, where do we found them, is the paracortex of the lymph node. We do found them in the spleen, but in the spleen where? It's in the white pulp, or you can say that it is periarteriolar sheath, and also found in thymus. So how does T lymphocyte take part in the immune mechanism or the immune system? I had drawn a diagram for your convenience. These are two cells. The one which I had drawn with the purple, this one is an antigen presenting cell, while this one is a CD4 T cell. So there is this present of this MHC class 2 molecule on the antigen presenting cell, which is composed of alpha chain and beta chain. So one point in the T lymphocyte is that the T lymphocytes, they are MHC restricted. MHC restricted. So what is MHC restriction? Not always, but up to 95% of the cases, the functioning of the T cell is MHC restricted, while in 5% of the cases it is not. So let's understand what does that mean. Suppose this is an antigen presenting cell, and it, with the help of this MHC class 2 molecule, attaches with this antigen and presents this antigen to the CD4 T cells, and the CD4 T cell has a T cell receptor. T cell receptor is composed of an alpha molecule and a beta molecule in 95% of the cases, while in rest of the 5% of the cases this may be a combination of a gamma or delta chain. Okay, so what happens when this MHC class 2 molecule bound antigen is presented to the T cell receptor with the help of this CD3 protein also, the first signal is transmitted, and this CD4 molecule which I had drawn here, this act as a co-receptor also. Okay, So this was the first signal which is transmitted, and then comes the presence of this CD28 molecule, which binds with the co-stimulatory molecule, that is CD80 and CD86, present on the antigen presenting cell, and this give rise to the signal 2. What happens when this signal 2 is not there? That might be due to either there is defect in the CD28 or CD80 cell. So what will happen if the signal 2 is absent? The T cell will undergo apoptosis after that. Okay, so with this we'll switch over to the T cell markers. So what are T cell markers? Let's start with CD3, which is a pan T cell marker, and other molecules are CD4, CD8. CD4 molecule is present on the cell to which we call as helper T cells, and CD8 positive T cells are known as cytotoxic T cells. These CD4 positive helper T cells, we can denote them with the sign T helper cell also. So this is further of two types, T helper 1 and T helper type 2 cell. So when this T helper 1 comes into action, it produces interleukin 2. We'll have a short discussion on the interleukins also. It gives rise to interleukin 2, interferon gamma, and just to memorize that it is involved in type 4 hypersensitivity reactions. And T helper 2 cells, they produces interleukin 4, interleukin 5, 13, and they also stimulates IgE synthesis, and they are associated with type 1 hypersensitivity reaction. Hypersensitivity reaction, will be having separate discussion on that. And CD8 cytotoxic T cells, as I had already said that these cells, they are MHC restricted. So CD4 positive T cells, they only recognize those antigen which are bound to MHC class 2 molecules, while CD8, this shows restriction for the MHC class 1 molecule. Okay. So what is the role of B cell? How does it comes into picture with regard to the immune system? First and foremost, let's start with the percentage it constitutes. It constitutes about 15% of the circulating lymphocyte cell population. And what are the sites where it is located? It is located in the follicles or in the cortical part of lymph node, and is also found in the spleen, the red pulp. And how does it act? What all receptors are present on the surface of the B cell? Like the T cell, it also has a B cell receptor, which is formed by the IgM molecule usually, and may be formed by the IgD, but it is very less often. And what are the markers, B cell markers? So we have CD10, CD19, 20, we have 21, 22 and 23. CD10 is also known as common ALL antigen. CD19 is a pan B cell marker. CD21 receptor is also an EBV, Epstein-Barr virus receptor. So let's just discuss how does this IgM acts as a B cell receptor. For convenience, I had already drawn the diagram. So this IgM molecule, it acts as a B cell receptor here with other molecules like Ig alpha and Ig beta present in the membrane, which altogether helps in sending the signal 1. And there is another molecule CD21 which along with the complement component participates in generation of signal 2. So we have two diagrams. This was a diagram which I had drawn to explain the T cell receptor, and this is the diagram which I had drawn to explain the B cell receptor. So if we'll just compare these two diagrams, we can make out that the role played by the CD3 molecule in the T cell is similar to the role played by Ig alpha and Ig beta molecule. Similarly, the role played by the CD28, it is similar to the role played by CD21 in the B cell molecule. Cells of the immune system. So we had already discussed the T cells and B cells. Next we'll be discussing the natural killer cells, the macrophages and dendritic cell. So T cell, B cell, we had already discussed. This is natural killer cell. So what are natural killer cell and how do they take part in the immune mechanism our body offers? These cells, they are also known as large granular lymphocytes. Or null cell or non-T, non-B cell. Class 1 MHC molecules, they give inhibitory signals to natural killer cells. So the natural killer cells, they attack and they cause apoptosis in the cells which have altered or decreased MHC class 1 molecule expression. Okay, these two, they were part of acquired immunity. Natural killer cell is a part of innate immunity, inborn, and it shares some markers with the T cell like CD2 also. Let's start with the macrophages now, or mononuclear phagocytes, and they are a part of reticuloendothelial system of the body. They process and they present the antigens to the T cells. So they act as antigen presenting cells also for the T cells. And the last one is the dendritic cell. Dendritic cells, they are also the antigen presenting cells, and these are further divided into two types. One is interdigitating dendritic cells and the other one is follicular dendritic cell. So the interdigitating dendritic T cells, the example of it is Langerhans cell, found in skin. Langerhans cell histiocytosis. Diagnostic feature: on electron microscopy, what you will get? Burbeck's granule. and the markers on immunohistochemistry, these are CD1A S100 positive. And next comes this one, follicular dendritic cells. They are present in the lymphoid follicles. HLA or MHC system. So what do you understand by the term MHC, major histocompatibility complex system? So it is a complex. It's a combination of related genes or related proteins which are encoded by a combination of gene code. So what are those gene code and what is the location of it? The gene codes are located on, just remember 6p. It is short arm of chromosome 6. Let me just draw a diagram, pictorial representation of it. This is a gene complex. This is a gene structure of gene present on short arm of chromosome 6, which encodes for multiple proteins which come under the complex known as major histocompatibility complex, and it is also known as human leukocyte antigen complex also, because it was first found on the leukocytes, on the WBCs. So this gene is composed of three related group of codes, composed of class 1, class 2 and class 3 group of HLA gene. So what do you understand by HLA class 1 gene? HLA class 1 group of gene. HLA class 1 group of gene, it comprises of multiple codes which encodes for protein, to which we call as HLA A, HLA B and HLA C, and these are composed of two subunits, alpha and beta. Okay, and the cells which express them are all nucleated cells and non-nucleated platelets. So depending on how the question is framed, you may be asked which non-nucleated cell express HLA class 1 antigen. So the answer will be platelets. Or the question will be, which non-nucleated cell does not express HLA class 1 antigen? So the answer will be RBCs. Okay, and one more thing, this class 1 HLA, as I had already discussed during T cell description when I was giving, during its properties I had already mentioned that CD8 positive T cells or the cytotoxic T cells are MHC class 1 restricted. So the antigens which are attached to, or the antigens presented by the antigen presenting cell in which MHC class 1 antigen is present, only those antigen presenting cells, let me just reformulate it, that suppose this is a CD8 T cell and it has a receptor for, this is an antigen, but this antigen can be presented only by the APC. This is an antigen presenting cell which is having this class 1 group of HLA. Class 2 group. It encodes for various proteins like HLA P, Q, R. And this is composed of again alpha and beta component, and these are found on the surface of the cells, which comes under the category of antigen presenting cells. That will include macrophages, the dendritic cells, the Langerhans cell or the interdigitating dendritic cells, and B cells and some T cells. So one more thing, CD4 positive T cells or the helper T cells, they are class 2 HLA restricted. That means the antigen presenting cells which have this class 2 HLA molecule, only those cells can present the antigen to the helper T cells. And one is HLA class 3 group. This is encoded by non-MHC genes in MHC region, and these are actually not histocompatibility genes. That means they are not involved in tissue rejection, not involved in tissue or graft rejection. So what all examples come under this? What all proteins which are encoded by HLA class 3 group of genes? These are some complement like C2, C4, tumor necrosis factor alpha, beta, HSP 7021 hydroxylase. These are the proteins which are encoded by the HLA class 3 group of antigens. Let's have a brief discussion on cytokines. What are cytokines? Because now and then you will be dealing with the molecules which are categorized under this broad term cytokines. So what is cytokine, or what are cytokines? Cytokines, these are proteins or soluble proteins which are secreted by cells like lymphocytes. So if they are secreted by lymphocytes, they are called lymphokines. If they are secreted by monocytes, they are called monokines. So they may be secreted by NK cells as well, and many other cell types which secretes these cytokines. So how do we classify them? Broadly, we categorize them into these following categories. The first class is interleukins. interleukin from 1 to 26. Second is interferon, alpha, beta and gamma type. The third one is colony stimulating factor. They may be of granulocyte monocyte colony stimulating factor, or they may be just granulocyte colony stimulating factor, or they may be just macrophage or monocyte colony stimulating factor. Okay. The fourth one is tumor necrosis factor alpha. Lymphocytes, I already told there is a lymphotoxin, but lymphotoxin is not lymphokine. Lymphotoxin is another name for tumor necrosis factor beta. Sixth is transforming growth factor beta, and the seventh one is platelet derived growth factor. Let's understand the role of these cytokines. Interleukin 1, what role does it play? One role it plays, it promotes synthesis of interleukin 2. The other role, it stimulates T cell proliferation. Interleukin 2, what does it do? It stimulates proliferation of T cell, B cell and natural killer cell. Interleukin 3, it acts as a growth factor for tissue mast cells and hematopoietic stem cells. So it acts as a growth factor. Interleukin 4, it promotes the growth of T and B cells. Interleukin 5, it promotes the maturation of B cells into the plasma cells. Interleukin 6, it also promotes the maturation of B cells and T cell as well. Then comes interferon alpha beta. They have antiviral properties. And then comes interferon gamma. This also has antiviral properties, but it also activates macrophages and it also enhances the expression of class 2 HLA antigens. Then comes tumor necrosis factor alpha and beta. They stimulate T cell proliferation and also interleukin 2 production. So interleukin 2 production, interleukin 1 helps, and tumor necrosis factor alpha beta, they both stimulate the synthesis of interleukin 2.